Hi, everyone. Welcome to Science with Spice Between Experiments. I am your host, Dr. Danielle Spice, molecular biologist and biotech scientist, and I'm super excited to have started this podcast where we're going to talk to real scientists about their science, but in an accessible and easy to understand way. In today's episode, I'm so excited to introduce you to Dr. Peter Serles, who is a postdoc at USC and Caltech. In this conversation, we go deep into nanotechnology, 3D printing, as well as stem cell science and brain organoids. He is an incredibly accomplished scientist and one of my dear friends, so I really hope you enjoy this episode. Hi, everybody. I am so excited to welcome Dr. Peter Serles to Science with Spice Between Experiments. Peter and I have actually been friends for a very long time, and I just want to say that, Peter, you are a freaking superstar. Like, literally. It's actually crazy. And I've honestly been so excited to watch your career grow over the last 10 years. I mean, we've been friends for longer than 10 years, but you've, been growing your career for 10 years.
Yeah, it's crazy to think back to us in grade one and how far we've
Oh, I
come since
know.
then. And now that we both end up here, I never expected to go down a PhD route and then just kind of fell in love
Right?
with science along the way. And it's been a great experience ever since.
Who would have thought those little kids back then? Look at us now. It's actually crazy. Okay. So, for our watchers and listeners, let me tell you why Dr. Peter Serles is so freaking cool. So, he received his PhD from the University of Toronto, but now he's a Schmidt Science Fellow at two of the best science institutions in literally the whole world, Caltech and USC. And he researches how nano 3D printing can be used to make nanostructured materials and bioscaffolds. I know that's like a big mouthful, but we're going to get into it in this episode. And he's looking how we can engineer these new materials from the size of the atom all the way to its actual application and learning how we can actually build these and design these materials at all of these different scales of length. And if that doesn't make sense to people, we're thinking like nanoscale, microscale, actual scale that we can see with our eyeballs. And he uses his expertise to tackle problems in aerospace, neuroscience, clean energy, and machine learning, which is crazy interdisciplinary and so amazing. So, Peter, how are you? How is life in Southern California?
It's good. Thank you so much for having me on. I'm excited to chat about some of my stuff. And as I was just saying, like I really just kind of fell in love with science and it brought me into all these weird places, these cool opportunities, getting to work in really interesting places and around the world. And I have to say that living in Southern California from Toronto is, a lot nicer to spend my winters down here. And also the US funding climate's great and just has been a really awesome place to be working for a couple of years now.
I mean, you've done so much work across multiple different institutions. You also went to France for a while. How has like the international side of being a scientist kind of informed your research and your life and maybe wanting to stay in California long term?
Yeah, I think the thing that really blew my mind is we think of science as this rigid process of, going through the steps and the scientific process, but there really is no one way to do science. And the more I worked now across 7 different labs in 3 countries, and you work with different people, different environments, different dynamics, both like cultural backgrounds and scientific backgrounds, and you just learn so much about how to think about problems a little differently. And that's been a really fun piece of my research, getting to work in California, in Texas, in France, in Canada, across a few places. It's been really, really fun to see how everybody thinks about problems a little differently.
I feel like, especially because you're like so interdisciplinary in your approach to science, I would imagine that, like, as an engineer working on biological problems in neuroscience, like the neuroscientists have a way of thinking about stuff, and you just come in and you're like, hey guys, let's think about it in this totally different way.
And that's the whole point is I was really lucky to receive this fellowship from the Schmidt family and the Schmidt sciences. And their whole point was we'll throw you in an environment you're completely unfamiliar with, go look, talk to neuroscientists, stem cell biologists, and see if you can kind of figure something out in that in between space. And it's worked out great. I mean, half the time I feel like a moron because I can't keep up with them. And then I remember I have my own things to bring to the
Uh-huh.
table too. And it's that really nice intersection.
I mean, I feel like along that line, like bringing, I bet if you had any type of conversation with them about like nanomaterials themselves, like the stem cell, like I'm a stem cell biologist, I'd be like, what on earth? So, with that in mind,
Yeah.
Can you explain to a non-scientist what is biomaterials, bioengineering? If you could explain your current project, the collaborative effort between Caltech and USC to a non-scientist, basically like a teenager, like how would you get across what you're doing and tell them why it's so cool?
Yeah, so I think the biggest thing I kind of got me really excited to work in this field and really dedicate a lot of my time to it is the fact that we are in such a cool place with stem cells right now. We have the ability to generate stem cells from anyone's body, doing the kind of reversal process from your blood or your skin or whatever your base cell is back into a stem cell, and then you can make anything with it. And this has unlocked so many cool ideas, so much that's up and coming where we can make small organ replicates outside of the body, or we can use them for different therapies, or we can test drugs on them and see how they respond. But the big issue is that when you grow stem cells in a dish, we're talking about like a flat piece of glass or plastic, and that's going to be very different from your actual human body. And when we think about this from an engineering standpoint, the biggest one is like the stiffness, where you might have megapascals, gigapascals is this piece of plastic, and kilopascals in your body. So, like thousands of times difference in
Yeah.
terms of what the stiffness would look like. So, how do we create the right environment to help these stem cells grow and prosper? And how do we kind of use that environment to turn them into different types of cells down the road, keep them healthy, like really improve that microenvironment? And as you were kind of mentioning, I start all the way at the atom level. I look at how we can
Uh-huh.
construct stuff in terms of like far smaller than you can see. Typically, we're working with a scale it's about 1,000 times smaller than a human hair. So
That's so small.
remarkable to be able to kind of pick and place atoms at that scale and then slowly build it up. You're much smaller than a cell even at that point. And then you build up this environment and it tends to keep the cells a lot happier. We can kind of put media in it, different drugs we can load into it to modify the environment. And so now we can really unlock even more with these stem cells that are already so great.
It's honestly so cool. I've always been so intrigued by the whole like physics side, especially of like development and differentiation. And I know that you have been really working a lot with brain organoids, which, are these small organ-like structures in a dish. And they have so much promise, but we still have a long way to go. And, you know, I'm I feel like your research is really trying to get us towards where we need to go with it. So how much do you think that your research is affecting the differentiation of these organs in a dish? And like, why is having like this type of scaffolding so important for them?
Yeah, it's a really kind of key piece of the message because we've gotten really good at forming these organoids, as they're called. And what we do is we just drop all the cells in a dish and we put them in a dish that kind of forces them to compact into a ball. And then we give them the right growth factor, drugs,
Uh-huh.
whatever helped guide their pathways. But what you end up with is this cluster of tissue that you're still missing a lot of these biological cues. You don't have the right stiffness, but you also the big problem I'm trying to tackle is that they don't have veins running through them. So they don't actually get nutrients into the middle. And over the
Mm-hmm.
course of, the several months you're working with them, the cells on the inside are a lot less happy and will die after about 45 days or so. So you end up with about half healthy, half less happy. And so what I'm doing is with this bioscaffold, we're pumping media through it. We're basically making artificial veins that we then can grow the tissue around and keep the whole thing a lot healthier. And so it's a really interesting problem between that nanoscale 3D printing and
Mm-hmm.
using stem cell engineering and kind of meeting them in the middle where we can build these really complex devices.
So do you find that I feel like the idea of getting nutrients to those like inside cells, like how easy is that to do? That feels like a very complicated process. Because I mean, it's easy to like aggregate or basically make clusters of cells. Cells, if you kind of like spin them around in a dish or give them the right kind of well in a dish to sit in, they'll make a cluster. Certainly stem cells, they love to do that. So how can we like I mean, maybe it's just like a dumb question. I'm like, how do you do what you do? Like, how do you actually like get the nutrients into these insides of these organoids?
It's a good mix between we have some of these engineering processes, like we kind of build this whole structure and we hook it up to a pump and a fluidic device. And there's a lot of physics that have to go into how you actually
Uh-huh,
shape
I
it and
bet.
how you get the fluid in there. But it's crazy because cells, as you mentioned, are so good at doing what they're doing. If you give them the right
Yeah.
environment, they're happy, they'll kind of form around it, they'll form all the right connections, they turn into neurons, they start to form synapses and connect to each other and talk. And you can image them and they're all flashing in sync, meaning they're all connected to each other. And so there's some parts that we really heavily engineer and some parts we just leave to biology. And biology is a really good
Mm-hmm.
engineer in itself. And so it's it's finding the right balance, not overdoing it on the engineering side, but also letting it kind of run on its own.
So I've always been really intrigued by this whole idea of like the assembly of these organoids and how much they're able to really recapitulate or really show the actual development of what's happening in the actual person. And, I was always really interested in my PhD in learning about neural development. And, you think about the development of the primitive spine as these morphogens, these molecules that can be secreted from these cells that cause different cell fate changes throughout the developing neural tube. And that happens across all of our developmental tissues with different types of these secreted proteins. And I'm not going to lie, I feel like I often never really think about it in terms of how the architecture or the extracellular matrix, like what is actually holding on to these cells, how that actually really informs their development. So, now that you're researching this and this field is becoming so much larger, how do you anticipate this field is going to like learn so much more within the next 15 years?
Yeah, I think it's really, we're at the tip of the iceberg. And as you were saying, there's so much more that goes on behind the scenes. All the support cells, all the connections it makes to other regions of the brain, everything that needs to kind of develop perfectly at the right time. And replicating all of that in a dish is extremely, extremely hard. So there's a
Like
lot of
how do
things to
how do we
think
even
about.
like have the hubris to think that we could do that in the first place? Like I love that we're trying.
And that's so I think we pick at it one piece of it at a time. So I'd say right now we're quite good where we can make maybe 12 different regions of the brain. You get spinal cord, you get thalamus, cerebellum. You can kind of specify exactly what cell types and what arrangements you want in a dish. And now what's really exciting about the past few years is now we're starting to piece them together. And so now you can make a connection between two different regions and study how the formation can be influenced by drugs or in different diseases that might affect one part of the brain or the other. And you can see kind of the cascade effects where if one piece gets damaged, how that changes the interactions with the other. So it's all of these synchronous cues you need. And there's a really cool work that came out last year where they did look at the full neural tube. And it turns out there's only a couple molecules, a couple morphogens you need to really signal the whole arrangement of it. And that's a great one to be able to do in a dish because now you can control if you put a little bit of this drug on this side, but not this side, it creates this kind of gradient, this change across the whole structure,
Mhm.
and you get more of what would be your kind of frontal lobe versus your spinal cord as it progresses along. So you can really control and tease things out in a dish that are really fascinating and kind of, in many cases, we learn from neurodevelopment, but in other cases, we can start to understand neurodevelopment better because we can do it in a dish. I'd say one of the biggest strengths of this technology is that we really don't know what happens in the human brain in a lot of these cases compared to, we're taking tumor biopsies all the time. And so it's a little easier to work with cancer cells outside of a human body, but it's really hard to get a hold of brain tissue. So if you can grow these small clusters of brain tissue in a dish, it gives you the first time you can really tease out these individual things.
That feels so important in terms of like neurodevelopment and developmental diseases. Like that's all happening in later stages of human development when we just, ethically, it's really hard to study that. So I feel like this type of technology is going to be so important.
Yeah. And that's, I think, one of the really cool ones. Because we had mentioned you can take blood cells or skin cells, go back to stem cells, and then convert that into brain tissue after. So, what we end up with is your exact genetic signature is being held up when you start to make these brain organoids. So, if you have someone with something like autism spectrum disorder, Timothy
Hmm.
syndrome, these things that are encoded in your DNA, we'll actually see signatures show up in the brain tissue that replicate that. So, it might be that the neurons start firing in a different coordination, or they might start firing at a different stage, maybe a week earlier or a week later,
Hmm.
depending on the neurodevelopment. And you can start to understand all of that with really high degrees of complexity. And it's so cool to be able to see that and to start to understand these things with really clear
Right.
resolution.
So I'm really curious, you mentioned the idea of like different drug situations and how that could affect development or even just replicating a human brain in a dish or a human spinal cord in a dish. For a long, long time, the only way that we've been able to really do drug testing for like safety and toxicology and like actual just like knowing how it's going to work from an efficacy perspective has been in animal models. And so now with these new emerging technologies that you have a huge hand in now, like how do you feel about this kind of transition maybe away from animal models, especially given that the US in particular has been really pushing for new funding for these alternative, potentially even better solutions to this problem.
Yeah, I think it's a really, really cool opportunity. And I won't go as far as to say drop all the animals right now. I think, I'll acknowledge the limitations. We're not quite there yet. But especially in terms of brain tissue, there's so many differences between what you would get in a mouse or a human
Uh-huh.
in terms of the proportions, the complexity, the size of the cells, even. For things like immune system, they're a little more similar. And so you can kind of screen drugs in a little more similar of
Uh-huh.
a way. But for brain, you really have no idea what's going on, and mouse can only give you the tip of the iceberg. So this is a case where you can get much higher fidelity from that. The other really exciting thing is the estimates. It's about 100 times cheaper to develop it in these organoids than it would be in a
Oh,
mouse model. So if you talk about that grand, $1 billion from concept to market is a typical number people talk about. Reducing that by 100 times means you could really screen a lot more drugs a lot faster and work with it on human specific tissue. So there's a lot of things there that are really promising about it. But again, as I mentioned, because right now we're still kind of limited to maybe one part of the brain at a time. You don't know
uh-huh.
how that affects other parts of the brain. You don't know how it would affect the liver or other parts of your body that might have downstream effects. So it's a tool in our toolbox that can give us a lot of new insights. It's not going to fully replace mice yet, but it's really exciting to see where it can go. And you had mentioned there was the FDA Modernization Act a few years ago. So
Uh-huh.
the FDA said you can use these organoids as your preclinical trial. And I think it was last October was the very first drug that got greenlit for a screening in people from these organoids alone. And
Yep.
then there's been a lot of things coming up. I know of some clinical trials that are starting based on some organoid models across the world. So it's getting there, and it's really
All right.
exciting to see where it's going.
I feel like I love the idea of, you're right. Like we can't anticipate toxic potential or, off-target effects of certain drugs if we don't have the entire field of organoids actually, being elevated. And that if brain organoids are really, really great, we still maybe need to think about liver or intestinal organoids to see if there are any of those like toxic effects if we want to eventually move away from all animal models entirely. I don't know. I think it's exciting. I agree. I don't know if we can move away from animals quite yet. I mean, maybe tough question. What do you think is like the number one thing that the field, from your perspective, needs to do to just put it like above and beyond to really showcase to these drug developers that this is the tech that they need to be investing in and that you guys can be the next frontier rather than continuing to rely on animal models?
Very, very good question. And it's a hard one, but I
Sorry.
also have, no, I think a bit of a plug for my own work because I
Yes,
would say
please.
the biggest concern right now is repeatability. Where if I were to take stem cells from two different people and try to make a brain organoid out of them, they'll end up very different. And even
Mm-hmm.
batch to batch, from, as you're passaging the cells, they get older, they start to change their behavior. There's a lot of things where it's biology is always has a high degree of variance, a lot you need to figure out within the lines, but we're not quite at the point yet where we're getting perfect repeatability, which means it's
Mm-hmm.
harder to understand the exact effects. And so that is one case where bioscaffolds, because we can now control the environment with a higher degree of precision, we can start to improve the batch to batch effects. And this is something we've seen in both like static scaffolds, where it's just a piece of material you grow it in, and with microfluidics and you're pumping media over it or around
Mm-hmm.
it, both of them are kind of pushing them in the general direction towards better repeatability. And that'll, I think, unlock a lot more in terms of how we think about the field.
I love that. And great plug. That does feel super important. I absolutely agree. Like being in biotech, like we think about repeatability all the time. And even at early stages, like if we can figure out how to make stuff happen predictably, even if it's in a preclinical model, like that's what we're looking for, right? So I think incorporating controlling the environment feels essential. So up until now, we really only just like talked about what you're currently up to. And for the longest time, though, you were not a stem cell biologist. Like, you weren't a biologist at all. You started out as an engineer. So let's take a quick step back. And now that you're a biologist who started as an engineer studying nanomaterials, now you're doing both. What would you say was the biggest hurdle in expanding your expertise like that? Because that just feels so daunting, like to just straddle these crazy different fields within science.
Yeah, they are very different. And I feel like the problems you think about and even the way you approach the problems is entirely different, let alone my knowledge base was just not there. I mean, I think the last time I had touched biology was in high school, and I decided
Oh
I
my
was going to work
gosh.
on the hardest problem I possibly could, try to use
Right?
my knowledge for the best thing I could find, and that ended up being in stem cell biology and brain organoids. But it really required me going back to even what is a cell before I could start to think about
Uh,
stem cell biology. But I was very fortunate. I was given the time and this fellowship to be able to take the time and really start at the fundamentals. And a part of that was, you're Googling every other word when you're reading a paper. You're really starting to kind of
uh-huh.
figure out the
I
field from
remember those days.
scratch again, right back to when I first started research. But
Yeah.
I've been really fortunate to work with some awesome people who have both been incredible resources, like really good when
Mhm.
I'm bouncing ideas off of them, trying to understand these things. And realistically, like when I have terrible ideas, they've been very patient. They've been kind of walking me through and helping me understand.
No bad
So
ideas in a brainstorm.
no, of course, but when you're also brand new to the field, you do have some bad
Yeah.
ideas. I'll admit
Yeah,
that.
maybe a
So
little.
maybe a
Yeah.
bit. So there was some things that they also came with when I was asking those bad ideas. It was because I was thinking entirely differently, and some of
Yeah.
them end up being great. That we kind of talk
Mhm.
through. Maybe it's not there, but maybe you're on the right path. And so it's been really, really fun to work across disciplines. I think the hardest thing has genuinely been the lexicon and like people talk differently. They don't
Mhm.
use the same words. They don't think about things the same way. And so just figuring out how to bridge that communication gap has been really interesting. But as a whole, I would say, it sounds corny, but research taught me how to learn and I learned how to
Yes.
learn very early. So now I can sit down with a paper and I can think about what's the critical information I need to get out of it.
Mhm.
How do I understand this in the broader context? How do I think about research? And while my fundamentals still aren't there, I'm not a neuroscientist by any means, but I'm starting to piece together what matters in these cases.
I mean, don't worry, you talk about it with a lot of authority. Like you sound good.
Okay, well, if I'm getting the words right, that's a good place to
Yeah,
be. But
definitely.
yeah, at the end of the day, engineering, you bring your expertise and you meet with a client or you try to figure out what are you
Mhm.
trying to build for, and you really tease out that problem with depth. And that's all I'm doing here is it's a different thing I'm trying to keep alive or trying to design
Yep.
for, but it's about understanding what are your constraints, how do you work within that space, what do you have to offer, and so
Yeah.
it translated really well in the end, and it's been a lot of fun.
I mean, do you feel like it was just your like sheer confidence in yourself? Like, was there any particular like person or feeling that you had that like let you know that you could do this?
I think it was in a case where I just said, if I'm going to do science, I'm going to try to do the hardest, coolest science I can
Fantastic. Yep.
and just
Uh-huh.
try to really dive in to tackle some of the biggest problems I can. And this was something I had kind of learned about before and realized there was this big problem, the
Yeah.
vasculature that needs to be tackled. And I have the perfect tool set to do so.
Yeah.
I had spent my PhD working in nano 3D printing and how to think about these nanostructured materials, and I could apply that with a slightly different spin and try to tackle these
Yeah.
bigger problems. I mean, thankfully, again, I'm working with really the best in the world, like really incredible teams that have helped to fill in the gaps that I lack. So it's all come together quite nicely.
So, like, was it just like, you mentioned that you figured out that this was something that you could really apply your knowledge and your expertise to. Was there something about brain organoids in particular that got you excited and made you want to like switch gears specifically towards that model? Or was it kind of just like any organoids in general and like trying to get perfusion or vasculature or getting that nutrient like into the middle of it that really made you excited?
I think had figured out organoids as a whole are just like an incredible technology, really cool and up
Hmm.
and coming, really something that I was just kind of got me out of bed excited, like really interested to see where it would go. And as I look through them, I mean, the problem that I was mentioning where brains are really hard to come by, we're really hard to study. There's such
Yeah.
complex systems. There's just so much to be done with them. And I said, let's try to target that because that seems like where, if I can get this working, that would make the biggest impact. And we are now in conversations now that we have kind of a working platform to be able to put it in kidney and put it in heart and try to use it in modify it for the environments that those need, but with the kind of same basis that we have started here.
Because I would imagine that the physical characteristics would be different across different, like the tension required in a kidney might be different than in a brain, and the perfusion requirements of, microfluidics going through an organoid would be different depending on the actual organ that we're talking about. So that feels really important.
Yeah, yeah. And it just becomes the same of what I was kind of saying. As an engineer, you just think, what are the problems? What are the
Mhm.
constraints? How do I think about this problem in a different context, apply the tools I have? And so it's been fun to even kind of start to expand it step by step. And even as you mentioned, like when we go between different parts of the brain, they need entirely different scaffolds. If you're looking at the
So
cerebellum
true.
versus the thalamus, like they have entirely different portions of cells, of neurons and their glia support cells.
Hmm.
So how do you modify your scaffold to support that? It's a very fun problem where you're tuning all these levers, you have all these tools, and what I just find so cool is that the technology is there. The nano 3D printing is now a reasonably mature technique, and we can design exactly what we're looking for at the nanoscale with full precision.
So let's dive into this idea of like the nano scale, like the real like engineering side of this, because I feel like that I remember when you were first telling me what you were doing in your PhD like years ago, I was like, oh my God, Peter, what are you doing? I'm so confused. Like, what is this? It literally blew my mind, and I was so excited that you were working on it. So like on this idea of nano 3D printing, like why is it so important for us to have to think about these problems at such a small scale resolution?
Yeah. So the biggest thing is that smaller is often different. And when we talk about materials, that can mean that stuff gets really strong at the nanoscale, or it can be really flexible, or it can kind of take on all these weird properties when you have these really small amounts of material. And so that's a property that we try to think about and use for engineering applications. And when we talk about biology, it's the fact that we can control stuff at the size of a single cell. And so we can tune that environment. We can really make that kind of exactly what we're looking for there. But what makes the 3D printing side so cool is that everything we make is going to be made from a bulk material that you probably cut or
Mhm.
melt or something, make it smaller in a way to shape it. And it's really hard to do that at the nanoscale with precision, to cut
I would
stuff in the
imagine.
right exact dimensions. And we're really good at doing this with transistors and computer parts, and that's the way that we typically think of nanomaterials. But that is often just like a sheet of material that you can kind of cut
Mhm.
holes in or like work within that domain. So 3D printing came along, and there was some interesting physics that were predicted back in the 1930s that say you can print with such a small volume if you got all the physics right and lined it up. And yeah, now we can do about 1,000 times smaller than a human hair.
Which is just crazy to me.
It's really crazy to be able to kind of see these things and you slowly build them up piece by piece. You can
Right.
only see them in an electron microscope when you really zoom all the way
Uh-huh.
into them. Otherwise, I'll get a piece of glass that maybe looks like it has a piece of dust on it, and I can tell there's 100 samples
Nice.
in that one piece of dust.
Oh
But
my
it's
gosh.
it's a crazy world to be playing in, and it's really fun to now use that for all of these really interesting applications.
So I know that like I mean when I think of like scaffolds, I think literally like scaffolding, like on the side of a building. Like I'm trying to build a house and I have all my, it looks kind of like a box and then another box and then another box. So, is that like lattice typical structure the best way of actually approaching these things? I know you're also working on some other stuff that is really looking at some brand new ways to have new shapes, or I don't even know if it's appropriate to call them new lattices. Like, correct me. Like, how do we know what shapes to do? Like, I don't know.
That's the magic question, is what is the right shape? And it's, again, as we were saying, kind of depends on every application and everything like
Okay.
that. So I'll give you two examples. One of my works in my PhD, we were looking at how we could make materials that are strong and light. Typically, when we think of materials, we've got styrofoam and things like that that are
Hmm.
super light but aren't very strong. And then you've got
Yep.
steel that's heavy, that's stronger.
Hmm.
And it's always this trade off between the two. But we figured out that if you can shape stuff at the nanoscale, where you get these super strong properties, like I was mentioning, but you shape them in the right way, now you can start to build bigger and bigger pieces of these materials with the right structure. And so for that one, we actually partnered with this really awesome group in Korea where they were able to use these new machine learning algorithms to tell us what shape to print. And so we kind of defined the problem in a certain way. We made it mathematically optimized, ran all these simulations and trained it all. And it spat out these shapes that we had never seen before. And when we actually made them and printed them, they were incredible. It's amazing what we can do with these technologies now to be able to kind of
Right?
optimize it. It all comes down to the math behind it, but it's a perfect problem for it. Because before the math would really break down. You would have a hard
Hmm.
time optimizing its perfect little bits, but the machine learning is so good at that. So what we end up with is now we can make this material that's as strong as steel, but as light as styrofoam at the same time. And it's incredible when we think about aerospace and all our planes in the sky.
Yeah, right.
It's as strong as the aluminum that's being used, but it's 15 times lighter than that aluminum.
And that feels so important. Like, especially even if you were like gonna transition that to like climate change or like anything like that, like so many things can be applied with this incredible technology.
Yeah, exactly. It's the fuel savings. If you replaced a kilo of aluminum with our material, you'd be looking at about 80 liters per year in fuel savings. And there's roughly 50,000 kilos to be replaced in a single plane. So there's a lot of potential for fuel savings, and that's carbon
Hmm.
dioxide emissions, costs, everything that would come along. So we were really excited about that work when it came up. And now
Yeah,
we're working
right.
on scaling it up, trying to make more and more of this material, seeing if we can actually build stuff out of it. And then the other example I would give
Yeah.
is when we think about our brain organoids, what is the right shape? And we've tried a few different
Oh God,
things. First, it was basically straws because it was easy to get the fluid to move through them. So
Yep.
just an
Okay.
array of straws. But turns out that they'll cells will align down your direction of your scaffold. They like stiffer things to kind of align themselves with.
Hmm.
So it turns out that what we decided was we went back to a CT scan of a brain and we actually could take that and use the engineering model of the veins running through a brain.
What?
We developed this kind of mathematical model that would roughly approximate this and shape all these new ways. And now we're printing with that same kind of branching structure and all the randomness that comes there.
That's
And the
amazing.
cells are actually a lot happier because we're not influencing them. This is what I was saying of
Yeah.
sometimes you over engineer it and sometimes you let them do their own thing. And this was a case where if we could remove the engineering influence, they
Yeah.
formed much more similar structures that you would get in the brain because we stepped back and let them do their own thing.
I mean, you're right. I feel like biology has had what, you know, 4.6 billion years to try to get to its like most optimized position at this point. Like, you're right. I think
There's so
taking
much we
away
can learn
that,
from it.
yeah, taking away that, like, I love that, like, over, like, thinking about what a great solution. What was there anything that like really made you think that a CT scan of the brain was like the way to go? Like, it was just like so obvious to you?
Yeah, I mean, it was a cool thing where you can go from CT scans directly to like a computer model and you can get all that with
Yeah.
like perfect precision. So we said, may as well learn what we can from nature. And that's a classic way people think about it, even in the materials sense that I was talking about before.
Hmm.
Materials in nature are great. Like the shell of a snail is so strong and so light because it's structured at the nanoscale. And that's honestly things like that are basically what launched this whole nano world because we realized when you just keep going smaller and smaller, nature has it right in a lot of cases.
Yeah, right? Like, I don't know how much you would know about it, but I remember like years ago, I learned about the like nanostructures in butterfly wings and how like crazy refractive index is for all the amazing colors that butterfly wings have. And I was like, this literally blew my mind. And of course, people want to try to replicate this because, like, that feels so important for biology and like learning from nature. Like, they've clearly figured it out, the butterflies.
Yeah. Yeah. It's so cool because it just comes down to how you shape the structure and the light bounce off it certain ways and you get
Yeah,
these beautiful shimmering reflections and it's something we can now do in the lab because we know those exact shapes and we can just print them and make them.
that's honestly so freaking cool. Okay, so I mean, you're clearly so incredibly accomplished. You've got like 40 different publications. You know, you've got some that are coming down the pipeline, I can imagine, very soon. I think it's really important for anybody listening and watching, and just like scientists and non scientists alike, to know that like we're just regular folk, you know, like we're just normal people. So, can you share with us, you know, outside of wearing your lab coat, what are your favorite things to do outside of the lab?
Yeah, I think that there's so much value in having that work-life balance and something that as I hope to be a prof someday and really move in that direction to run my own lab and something I'm always going to make sure to instill because some of my best ideas come when I'm not in the lab. And that's without a doubt, I would actually say like all of my best ideas have been out on a run or out on the ski hill
Hmm.
or whatever I'm doing with my time, then my brain's working in a different way. And so I try to really prioritize that. And so I'm going for a triathlon this year and training for that.
Oh,
I love to ski and I've skied in uh, 3 continents now and 40 something places and I love exploring new places. And so it's a lot of fun to be able to use my time away from the lab to really
hmm.
unwind and really relax. And there's as much as science gets me up out of bed in the morning, there's a lot more to life than science. So you also have to make sure you have that balance. And it's a great way to think about life too.
No, I agree. I feel like I don't know what it is about like moving your body. I don't know. I feel like it's like you're almost because you're in like a totally unrelated flow state, like doing a run or skiing or doing something else that like is almost like the background tabs are able to like figure out ideas while you're doing this other flow state activity. I love like moving my body to like think about science and then just
Yeah.
come back to it.
Just yesterday, I was working on a coding problem for maybe 8 hours and couldn't crack it. And I went for a run this morning and it came immediately.
Fantastic.
That was just what I needed. Yeah,
Oh my God, then you have to say like I should have just gone for a run yesterday.
yeah, exactly. That
Like
would have been more
halfway through.
productive.
Well, it's so hard to like step away when you're like so in the problem though. So, I mean, right now, like you're still in your postdoc, but you know, you just mentioned that you're potentially interested in developing your own lab. I mean, I feel like given your breadth of expertise and all the different applications of this technology, like is a, you know, founding of a company in your future or just a lab for now? Like don't put the cart before the horse, Danielle. Like,
I'm keeping my options open. I think the
okay.
idea of running my own lab, whether that's in a company, whether that's in a university, whether that's in a national lab, or working within a bigger infrastructure, I'm open to see where it goes. And one of the great things about this profession is that it's kind of a global case where you can look into different
Mhm.
countries and different styles and funding atmospheres. And so we're keeping the options open to see. We're in the middle of interviewing and discussing with a few different cases. We'll see how it all comes together. But I think what I'm really interested in is I have a lot of things I'm really excited to work on. And the freedom to work on your own ideas is such a privilege and something that you get in, I would say, a lot of atmospheres. So being able to really think about how I can set that up, how I can work with really great people who can work on some of these ideas, push back and push me to think about
Mhm.
ideas differently. It's going to be, it takes a bit of sculpting and a bit of searching, but I'm excited to see where the next steps go. For now, I'm very happy in Southern California working with really great people and on some really big problems, but excited about what the future is going to hold.
And I am so excited to see where you're going to end up next because all of this sounds so, so amazing. So, with that, I want to thank you so, so much, Peter, for taking the time between experiments to talk to us.
Thanks for having me on. Hopefully, I was able to share a little bit about the nano world and all of its
Oh, yeah.
big impacts. And there's a lot of really fun things coming down the pipeline. It's cool to now start to see how stuff gets out of the lab and gets into the hands of helping people. And it's why we do the science we do. It's a fun place to be, and it's a privilege to get to work in it. So I'm glad I could share some of that today.
Oh, absolutely. Thank you so much.
Thank you.
I had so much fun talking to Peter, and I hope you guys really enjoyed that conversation. Don't forget to hit subscribe on YouTube or wherever you get your podcasts, as well as you can find me on Instagram at Science with Spice and TikTok with the same handle. Until next time.